This is the lecture for Lab 2 focusing on enzymes. So, what is an enzyme? Well, it turns out an enzyme is A and B. It is a catalyst and it is a protein. At least for our class, enzymes are always proteins and they are catalysts. So, they are proteins that serve as catalysts. Now, there are other things that can be catalysts, lots of other things that can speed up chemical reactions, but in living things, the main, almost exclusive type of catalysts are enzymes. That's why we're focusing on enzymes. Now, catalysts are chemicals or things like enzymes that can increase the rate of reaction, can make the chemical reaction occur faster than it would all on its own. And catalysts are not changed by the reaction, so that at the beginning of the reaction, the catalyst is the same as it is at the end of the reaction. And that means catalysts can be used again and again and again and again, which is nice and convenient. And again, overall, they are speeding up the chemical reaction by decreasing the energy of activation needed for the reaction. But they don't change the basic nature of the chemical reaction. It's still the same process overall. So, for instance, here we have sucrose, common sugar, and over time it will break down into glucose and fructose. Now, on its own, it takes sucrose, thousands of years to break down into fructose and glucose. So it has a very high activation energy that has to be overcome before the chemical breakdown occurs. But if we add an enzyme to catalyze this reaction, then that enzyme will cause the sucrose to break down into glucose and fructose into less than a second, super amazingly fast. So the enzyme has lowered the inactivation energy quite dramatically, speeding up the breakdown of sucrose into glucose and fructose, which is awesome because then we can eat it and get the nutrients out of it. So enzymes have an active site. The active site has a specific shape. That shape will fit a specific substrate, some specific substance of the correct shape, only thing that can fit into the active site. When substrate binds to the active site, we now get the enzyme substrate complex, and this is when the catalyzing of the reaction occurs. This means that the shape of the enzyme is critical for its function, and it is often referred to as having a lock and key function, which means the enzyme acts as a lock and only the specific key that can fit in the lock will open it. Substrate, perfect shape, fits into the lock active site of the enzyme. So here in this example, we have two substrates that come in and bind to the active site. When they bind to the active site, we get the enzyme substrate complex. Catalyzation of the chemical reaction occurs and out pops the product, and there are two products in this case, product C and product D. So reactants A and B go in, chemical reaction occurs, catalyzed by the enzyme, giving us products C and D. Again, the products are over here are pointed to by the arrow. The arrow always points to the products. Enzyme naming. Most enzymes end in the letters A, S, E. Group got together, decided that is how enzymes must be named. So if you ever see something that ends in ASE, then it is an enzyme. Phosphatases are enzymes, remove phosphates. Synthetases, enzymes, cause dehydration synthesis. Amylases, enzymes that break down carbohydrates. There are enzymes that were discovered before this convention was developed. So there are enzymes out there that do not end in ASE, but if it ends in ASE, it is definitely an enzyme. So, things that are important is figuring out, measuring the enzyme's activity, how quickly it causes the substrate to be converted into product. And we can do this by measuring how much product is formed over a given period of time. If there's more product formed, that means the enzyme was faster. Less product formed, that means the enzyme was slower. And there are various things that can influence this temperature, the reactions occurring at pH, the amount, the concentration of the substrates, and the amount or concentration of the enzyme. So temperature, in general, on this chart we see here, we have enzyme activity. So the higher up it is, the faster the enzyme's working. Lower down, the slower it's working. And then temperature, 10 degrees, 20 degrees, 30 degrees, and so on. So temperature is increasing as we go to the right. So at low temperatures, the enzyme does not work that well. We don't get much chemical reaction. But as you raise temperature, you will speed up the chemical reaction until you get to the ideal temperature. That temperature, the enzyme works the absolute best it can. That's why we got a happy face. And then if you continue to increase the temperature, the enzyme loses activity until eventually you boil the enzyme, destroying the enzyme, and there is no function. So again, increasing temperature will increase activity until you get to the ideal temperature, and then further increasing temperature will decrease activity until you denature or destroy the enzyme. So denaturing enzymes. You take an egg, you fry it, the egg changes colors. That's proteins in the egg's whites of the egg getting denatured. And after you do that, you can't undo it. Denaturing a protein is permanent. You can't unfry the egg. All right, pH. pH works in a similar manner, where here again, enzyme activity, faster is higher up. Acidity, in this case, it becomes more basic, further to the right you go. Now, various enzymes in our body work at different pHs, so different enzymes have different ideal pHs. So, for instance, pepsin has an ideal pH that is very low, very acidic, because it functions in the stomach. Salivary amylase is kind of neutral, it's in our mouth, and trypsin is in the small intestine, so it's somewhat basic. So as you increase pH, you'll increase activity of the enzyme until you reach its ideal pH. Further increasing the pH will decrease enzyme activity, and at the extremes, super acidic or super basic, you will destroy the enzyme, it will denature and stop functioning. Salivary amylase, same idea, increase pH, you increase activity till you reach the ideal pH of around 7. Further increasing pH will decrease the activity of the enzyme. Amount of substrate. The more substrate, the more reactant you add to the enzyme, the faster the chemical reaction will go. So as we go to the right, we're increasing the amount of substrate. However, there is a limit, a saturation. So increasing the amount of substrate is relative to the amount of enzyme. So if you add a little bit of substrate, it'll go a certain speed, add more, it'll go even faster based on the number of enzymes. But once you're providing so much substrate that all the enzymes are functioning, you are no longer speeding up the chemical reaction. So again, as you increase the substrate, you increase the speed of the reaction until you reach saturation, at which you cannot make the chemical reaction go any faster. Amount of enzyme, as you increase the amount of enzyme, you speed up the chemical reaction as long as there is excess substrate. So the more substrate you, as long as there's plenty of substrate, infinite amounts of substrates, then the enzyme, as you increase more enzyme, add more enzyme, it'll keep going faster and faster and faster. In a perfect world, you do not have an infinite amount of substrate, but in our example with these robot enzymes, you keep adding more robots, keep adding more enzymes, there's plenty of substrate, so it keeps getting faster and faster. Cofactors and coenzymes are structures that some enzymes need to work. The cofactors are often minerals, inorganic, they have to bind to the enzyme to get it to function. Coenzymes, often organic like vitamins, again, they have to bind to the enzyme to make it function. Some enzymes have cofactors and coenzymes, others do not. So if there is a very little bit of the cofactor or coenzyme, it will be fewer enzymes will be functional, so they will be slower. But as you add more cofactor or coenzyme, the enzymes will become active until you have all of your enzymes active. After that, adding more cofactor or coenzyme will have no effect. So again, increasing the concentration of the cofactor or coenzyme increases the chemical reaction speed, the rate of the reaction, until you reach saturation, until all the enzymes are bound to cofactors or coenzymes. Again, only some enzymes need these cofactors or coenzymes. All right. That is where we'll stop for right now.